I'm Arlene Montalvo. This is the Riverside San Bernardino chapter of the California Native Plant Society. This is our regular third Saturday meeting and program, and we have quite a few people in the room today, which is exciting. And people are almost fully seated. There's still some people signing in here. We have about 20 plus people online as well, and about the same number in the room. So this is a great turnout today. So I'd like to welcome all of you and thank you for joining us. When the speaker is talking later, those of you online who are listening, please be sure to write your questions in the Q&A box?
Yes.
Yeah, in the question and answer box. We will try to follow the chat, but I'm not sure we will be successful. It depends if my computer stops running because it's not plugged in. And then also, I just want to make a few announcements before we get started with our great program, which I'm excited about. We have President Biden signed the national monument onto the making the Coachella. I'm sorry, Katie, help me. The Chuckwalla National Monument and the Satilla National Monument last week, and Katie Barrows, who's co-hosting this webinar from remotely, attended the White House signing, as did our executive director, June Bando, and a couple of our conservation people, like Nick, some of our staff. And it's just very exciting that this actually happened, and we're just, it's just wonderful. So anyway, thank you, Katie, for all the hard work you did. And then also we have our season of programs coming up in February. On February 15th, Chris McDonald will be providing our program. It was going to be a weed identification workshop, but we've had to shift that because we've had no rain and the weeds are not growing. So instead, he's offering a different program, which will still be exciting. It will still be an in-person only program. He's going to be talking about drought adaptations in plants and native plants, and we'll have lots of examples of the different kinds of adaptations that they have present in the room. So that should be a fun meeting and workshop. We're still arranging our March meeting. It might be a program on using the updated CalScape and also CalFlora. CalFlora has all kinds of cool tools in it. So we might spend some time, doing a hybrid program, messing around with CalScape and CalFlora to give those of you who don't really know how to use those tools real effectively, give you a better handle on it. So that could be a very powerful thing for you. Then in April, I think we're having a horticulture program about, I'm sorry, I'm not remembering her name right now, but we have somebody who's offered to give a program on starting her native, yes? I don't know what, we're just having people. Oh, okay. Sorry. Yeah, I don't know what's going on. So, anyway, where was I?
Horticulture
April,
program.
yeah. Okay, so it's I think she'll be talking about starting her garden from scratch and what she went through, in planning her garden and installing her native plant garden. And how she combined that also with some things like fruit trees on a large property that she has. So that could be very interesting and very interactive. In May, we're really lucky. We're going to have Nick Jensen, he's the director of our conservation program, his staff at CNPS. He will be coming here and he will be introducing us to the really great conservation toolkit that CNPS has put together to help people understand, what we go through and what the steps we take when we're trying to ensure that our land is developed in a way that's more friendly to the environment and to protecting native plants and vegetation communities. He will also be talking about the Wildflowers of California. It's a beautiful book. I'm just going to grab it. He'll be talking about, see if it will show up. Trying to get it to, if I hold it to my face. No, it's not gonna, I can't get it to work because of the background. Anyway, it's a beautiful book, Wildflowers of California. Many people from CNPS helped put this book together with beautiful photographs of many of the common wildflowers throughout California. And I think there will also be books available for sale that day and for signing. Get rid of that. And then in June, I think what we're going to do is be at Heaps Peak Arboretum, where Gina Richards will give us a tour of some restoration projects that they've done up there. So this will be an in-person only one, but we're going to actually go somewhere else. We're going to go into the San Bernardino Mountains, look at this restoration site, have, a little lecture about it, and have a picnic. So it'll be a very different sort of meeting compared to what we normally have. So that will be our June meeting. Exciting news, we have a field trip chair who is going to be organizing a bunch of field trips for us. I can't really tell you what they are yet. He's got lots of things in mind and ideas are coming in. If any of you have some ideas, be sure to contact us. Places that you've seen that would be really great to go, especially in a drought year, okay? We will have a different focus than looking at lots of wildflowers, I think, for our field trips this year. I think that's it for the announcements right now. So what I'd like to do is introduce Chris Coshland. She is going to be giving our program on bryophytes and lichens, which are wonderful creatures. Chris has a very broad background. She's worked for various federal agencies in a biologist and botanical capacity. She's currently a natural resources or cultural and natural resources manager at March Air Force Base. She's just full of all kinds of knowledge, environmental. And I think I'm just going to go ahead and have her come up and give the presentation. Oh, she may also be joining our meetings and giving us updates about what's going on with bryophytes in California each time. So I think we just need to
not open it yet. I'll open.
Okay, great. Thank you.
Thank you. So what I didn't put on my presentation is that CNPS has its own chapter for bryophytes, and I've been tasked with representing the Inland Empire in that chapter. So I am the representative for Bryophyte chapter for Inland Empire, but I'm also lichen, so I'll be representing us anyway. Then okay, how's that?
It's not full screen.
That's the fullest we can get, remember?
Okay.
We couldn't. Okay, so this video is just a speed up video of lichens being hydrated. I just love it. The cool thing about it is you can see it's like a whole nother world. I mean, lichens move very slowly, so you don't actually see them move, so that when you speed them up, you can see just like plants, they respond to the light, move around. So what we're going to talk about are exactly what are non-vascular plants, mosses, liverworts, hornworts, and lichens. And the most common, we're going to touch on the most common species of Southern California, and just a brief on a current project I'm working on. And extra tools for anybody. So a little bit about myself. I worked for BLM and Forest Service for 20 years as a botanist on 6 forests in 3 states. I was a district botanist, managing all botany and habitat related projects. These are just some of my seasonals. The bottom left shows some of my seasonals as we're doing a rehab project, transporting native plants into a rehabilitation site. And on the right, and these were actually, these 2 on the bottom were in Oregon. And then on the right, some other seasonals just posing. So I also did a special project where all of us district and forest-wide botanists got together to help another district botanist in Oregon to relocate this rare moss. It's a dung moss. It only grows on dung. So we wanted to move it off this area that was proposed for a timber sale. And they were proposing a temporary road there. And currently I work, like she said, but it's March Air Reserve Base, not Air Force Base. We're just wicked stepchild of the Air Force as a reserve base. As a natural cultural resource manager. This picture is just us doing fairy shrimp surveys. And lastly, I'm currently the curator at UCR for Bryophytes and Lichens. So what exactly are non-vascular species or plants? Non-vascular plants contain no true vascular system like regular plants, such as xylem and phloem. Instead, the tissues of the vascular system are in a tissues that, sorry, that's supposed to be non-vascular system, are of a simpler form. Water is directly absorbed through the cells themselves instead of going up through xylem and phloem. So today, non-vascular plants are represented by algae, bryophytes, and lichens. Of course, we're not really going to talk about algae today. So in the fossil record, 2.7 billion years ago, cyanobacteria showed up in the fossil record, was the first light-producing species that produced oxygen. Cyanobacteria was around for a while, but it wasn't actually taking the carbons out of the atmosphere to convert it through photosynthesis to produce oxygen. So we had the first cyanobacteria that finally did this. 1.6 billion years ago, red algae appears in the fossil record. I think I have the wrong presentation open. I'm sorry. I just realized I have the wrong presentation on. This is another one. Sorry.
No
Yeah,
worries.
this is not it. Okay, let's try this again. Okay, so this still not it. Hold on.
You stopped you stopped the sharing?
Yeah. I apologize. Okay, this is it. Okay, so where's my Teams go? Zoom. Where did, not Teams, Zoom. Where did my Zoom go?
It's there. It's on.
Okay.
Yeah.
Not showing up here. Oh, there you are. Okay. Still not showing up. This is it.
Okay. So I'm not seeing it yet.
There it is. Okay, sorry about that.
You do that
Hide.
control alternate shift H to hide that stuff once you're ready.
Okay. All right, sorry about that. Go ahead. All
Hopefully
right.
that stripe in the middle will go away.
There's a stripe in the middle.
Yeah, it went away. It went away. We're good.
Okay. All right. So just a brief, we're going to talk a little bit about the evolution of these land plants. This is kind of an artist rendition of how the first photosynthesizing plants were algae and how they evolved from the sea or fresh water onto land and became plants. So this is just a real quick first steprophyte algal diversity 600 million years ago, and then boom, we have trees. It's just a quickie, but here's a nice little artist rendition. This is at an actual museum where they recreated what it was like 4 point something million years ago when plants were starting to evolve onto land. These white things are actually large, what they think might be fungi. They're now questioning whether they are, but they stood really high. They're really huge. Oh no. Okay, so what are bryophytes? The earliest plants are believed to have evolved in areas like the coastal mudflats in the Devonian 410 million years ago. The first non-vascular species were Bryophyte sporangionites. These are just kind of pictures of them. You can see these right here are the Bryophyte sporangionites. Then you also have the Cooksonia, Rhynia, Zostrophyllum, and the Drepanophilus. And here's the actual fossils of these species. So these are artist rendition of the Bryophyte sporangionites, and then these are the fossils right here. And then here's Cooksonia, very interesting. Some Rhynia, there's some more Rhynia. Zostrophyllum, looks like almost like a grass, but these are the first plants, or actually the oldest known plants that we know of. So in the fossil record for bryophytes, I think we were going through this. So you have cyanobacteria, which was producing oxygen, red algae, and then 1 billion years ago is where you start getting into the charophytes and chlorophytes, which is a freshwater algae. And then between 473 to 471 million years ago is the oldest known fossil in the oldest known fossil record of liverwort spores found in Argentina. There was 5 genera, which shows plants were already beginning to diversify. 419 to 393 million years ago, you got hornophyton, which is a possible precursor to hornworts. And this is an artist rendition picture of it. And then 407 million years ago is the oldest known terrestrial flora symbiont with fungi called Winfrinatia reticulate, which is the oldest known terrestrial lichen found in the Rhynie chart, and Spongiophyton nanum, a putative lichen. Thought I had pictures in here. It's not. So let's look at break down what bryophytes are a little bit more. We have moss, which represent general moss and sphagnum moss. And over here in this diagram, you can see it has a leafy section. This is all called the gametophyte, and then the sporophyte for the moss. In liverworts is very similar. You have leafy and then you have thalloid. And then thalloid can be broken down into 2 different types, simple thalloid and complex thalloid. So this is a gametophyte of the liverwort with their sporophytes. And then you have hornworts, which we'll get into more. So they're considered, I've mentioned this before, they're considered non-vascular because they don't contain a true vascular system. Instead, they absorb straight through the cells. And if you look at a vascular system, you'll have these vascular bundles, which is where the xylem and the phloem go up and down the plant stem, right? Moss don't have that at all. They have kind of a rudimentary central core, but it doesn't transport water and it doesn't transport nutrients up and down. Instead, they absorb completely through the cell, and each cell has its own chloroplast. So it is going through photosynthesis, just in a very rudimentary way. So this picture right here actually is the same moss. Moss, because of this not having, not being a full vascular plant, it's able to completely dry out and desiccate, and it can dry out and desiccate for years. As soon as you add water, it comes right back to life without causing damage. Okay, let's see if oh, here's an example. So this right here is how fast some species of moss can actually hydrate when you add water. This is real time. This particular, that's all. That's just another video we don't need to go over. This is a picture of a sporophyte releasing spores. Sometimes you could just go right up to it on your patio if you see any spores. And if they're mature, you can just shake them and you can see them. Especially in the nice wind. All right, so bryophytes have a sexual habit, I guess you'd call it, of having sex and producing asexually. So here's an example of two gametophytes on the same plant. You can have males at the very top called antheridia, and then you have females at the top called archegonia. And sometimes you can have both sexes on one plant entirely. The point is, they mostly reproduce by water or rain. So if the sperm is ready on top and the rain hits that, it spatters the sperm, and all it has to do is land on the female, which it goes through this tube right here to produce a new species. So here's some actual pictures wrapped up of some of the male and female parts. And then these are just some examples of how different some of the moss gametophytes look like. They could be branched, like down here, which is called a pleurocarp. This is a pleurocarp, and then these are acrocarps, so they're unbranched. The difference between a pleurocarp and an acrocarp, besides the branching and not branching, is that the sexual reproductive structure usually is on top of the moss. The spores come out at the top of the moss. And on pleurocarps, the spores will come off the main stem of the branched moss.
Why the gray box showed up again? I don't know if you can try that control alternate thing, hide again.
The gray box? I don't see a gray.
I know you
box
don't see it,
online.
but you can see it.
Can I see which gray box?
It looks like
weird.
it popped out after the video.
Okay, so I'm asked to reload this because there's a gray box showing.
No, I think, remember how you hid the stuff at the top before with the control alternate shift? Oh, it went away. Oh, it went away. It's
Oh,
gone.
I just minimized it and put it back.
Oh, no, it's
I don't know what it is.
Yeah.
You go back one where the man was old.
Oh, that was my next one.
Pardon?
That's my next one. I haven't gone there yet. I went back because I went too far. So
Maybe try.
it's good. When I do that, it goes away.
Yep.
And then it comes back, right?
No, it's got, well, now it's back.
Yeah.
Yeah, so try hitting this gate and the other stuff's gonna come up.
Mhm.
Now try hiding
They're going down to the hide.
and then you're gonna hide again. Let's see if it. Before one, hopefully that.
Did it
one
work?
In the middle will go away.
Yep.
It did work.
Well, don't worry, just keep going.
Oh,
Sorry.
sorry.
Don't know how to get rid of it. Yeah.
All right. So sphagnum moss is another kind of moss. It's a lot more primitive, older in the evolutionary terms. It mostly inhabits more acidic environments like peat, where peat moss is produced, where peat is produced, such as bogs and fens. So if you could look, here's a example of what a sphagnum moss looks like. It grows really fast, so it produces these long gametophytes, and because of that, the acidic environment has no time to actually decay the moss. So what happens is it grows really fast. The acidic environment is kind of slowing the anaerobic decay of that moss, and it builds up, and that's actually what builds peat. So peat is very unbroken down moss over thousands of years. It's an example. Another thing about sphagnum moss, which is interesting, is each one of these sporophytes comes from a different father. So how they produce is completely saturated in water, and the sperm will float around in the water, gets to the female. The female will put out several of these sporophytes from different fathers. Interesting. So bog water is as sour as 3 to 4 in the pH scale, and fens, which I did not bring up here, go from 4 to 7. So they can be neutral to acidic. And a lot of these species, like sphagnum moss, are specialized for those acidic environments. And here's just some really cool different pictures of different types of moss. Also, peat from this environment can accumulate up to 10 meters in depth. So I was doing fen inventories up in the San Bernardino National Forest, and put my auger down into one fen that I found and pulled up 40 centimeters, pulled up a piece of wood that's never fully decayed after 40 centimeters deep. And they say that 40 centimeters is approximately 30,000 years old. So I was holding a 30,000-year-old piece of wood. It's kind of cool.
Somebody in the chat is saying that box, it says it is the participant's window on the presenter screen. You can shrink it by hitting, I think it says the minus, but I'm not sure. The minus sign,
Is there an underscore?
an underscore, oh, by hitting the underscore, I don't see. Maybe in Zoom, but I don't see that. Yeah,
That was in
I'm not
Zoom,
sure
wasn't I?
exactly what they mean, but.
Have a presenter screen.
Where's my Zoom? I lost my Zoom again. When did I do that?
Don't know.
Rod saying not underscore, but on the black bar on top there are. Oop,
Gone now.
For now.
It's gone for now. I think it's okay.
Okay, so
Yeah. It says not underscore, but on the black bar on top, there are several icons, and one is a underscore, or drag it to the side, but I think you got it. It's on the side.
oh good. I don't know how I did it, but
It's dragged over to
all
the
right.
side.
I'm just skipping ahead to get
Yeah,
back to where I
we're
was.
good.
All
Thank
right.
you. Okay.
Still gone?
There's another one that showed up that it'll go, okay, we're good. We're good.
It went away?
We're good.
Okay. All right. Uh-oh. Okay. All right. So let's talk about liverworts. We kind of we briefed on the moss, so we're going to talk about liverworts. These are just some examples of the different types of leafy liverworts. They could be, they can look like moss in some cases. They can have some crazy looking leaves. These are actually very miniature. You can only see these in the microscope for the most part. Some of them will, like this one right here has this right here is one leaf. So it's split almost all the way down. So then you have like, 3 months, you can have several of them. Leafy liverworts are amazing. Simple thalloid liverworts is one of the thalloid ones. And it's called simple because it's only 1 cell deep. So if you put it under a microscope and a slide, you should be able to see right through it, the 1 cell. And that's the difference between the simple thalloid and the complex thalloid. So these are just some examples of some simple thalloid liverworts. They come in all different shapes and sizes. And right here, you could see a sporophyte to one of them right here. And then complex thalloid liverworts. This is the one that has multiple cells thickness. This particular one is common worldwide, Marchantia polymorpha. And the cool thing about it is, if you look at this picture right here, this is the male, and it looks like an upside down umbrella. And right there is where all the sperm sits, is right here. These are the females, these palm tree structures. So what happens is rain will splash this, the drop will land onto one of these palm tree structures, and within those fingers, that raindrop just sits there and fertilizes that female right there. So that's what those structures are for. And at the same time, this one also does asexual reproduction. So this is what these little gemmy cups are for. And if you look here on this female one, it's also doing asexual reproduction, same time, on the same plant. So here's a couple more. This one at the very top, Lunularia, is pretty common in like throughout Inland Empire and people's lawns. It becomes a weed. And this one, you can see these are the gemmy cups, so these are in half. And then these are the archaeogoniaphores for these other ones. Then you have Rixia, several species of Rixia that we have here. These are just a couple examples of some of the ones that we have. This one's really common, these ones with the hairs. And then you have one that is very cavernous. It's actually called cavernosa, Rixia, cavernosa. Yeah, all different shapes and sizes of those. Now, hornworts are named because they look like horns. Obviously, they're very rudimentary. You can easily distinguish them in the field. However, without the horns, sometimes they do look like liverworts. They look like the thalloid liverworts. This one up here at the very top was one I took in Oregon. It looked like grass. But unless you knew that it was a hornwort, you know, you could see the thallus on the ground right here. And the tips turn dark because this is where the spores come out when they're mature. Here. So without horns, this very much looks like thalloid liverwort. But in the field, if you have a hand lens, you can easily distinguish it because it's one chloroplast per cell, and they're so big, you can actually see it with a hand lens in the field. So you can instantly be like, oh, it's a hornwort, not a liverwort. All right, so lichens. Let's get into lichens. Lichens are a symbiotic organism. They are composed of a fungal partner called the mycobiont and one or more photosynthetic partners called the photobiont. So here's an example of that. You can see that the algal cell, the photobiont, is immersed within the hyphae of the fungi, the mycobiont. So if you take a cross section of a lichen, you'll have, in most cases, you'll find an upper cortex, which is kind of like a condensed hyphae of fungi that protect it. It's like a skin on top, and you'll find that almost in all of them, unless it's E. cordate. In cyanobacterial lichens, you'll see cyanobacteria scattered, mixed in with the hyphae. In some cases, you'll have a layer of algae, and then you'll have a layer of the hyphae from the fungi called the medulla, like shown here. So there are about 17,000 species of lichen worldwide and 5,618 in North America alone. Both photobiont and mycobiont depend on each other for survival, food, and protection. Fungi harvest light energy from the algae. So they take the nutrients that the algae is producing, or cyanobacteria, which harvest light energy from the sun and generate their own nourishment in the form of carbohydrates, fats, and proteins. Fungi uptake the nutrients that could never receive otherwise. On the other hand, algae and cyanobacteria cannot survive outside of an aqueous environment. So without the protection of the fungi, it couldn't survive on that branch by itself. Once it's dried out, it's dead. They break down really easily. So evolutionarily speaking, we don't know how that relationship even started. We do have comparisons of what we do know, which is the association of fungi with plants. Plants, and especially trees, will have these roots, but then they'll have mycorrhizae fungi, which spread out even farther, take up more nutrients for the tree, and in exchange, the nutrients that the tree is producing will also further feed that fungi. So let's go on the history of it. So the Earth right down here was formed 46,000 years ago. 4.6 billion years ago. Let's just talk about 4.28 billion years ago, iron oxide tiny filaments called knobs and tubes were found in the Cambrian rock layer. I'm trying to get rid of my things popping up here. Okay. We don't know if it was some type of life form whatsoever, but since 3.5 billion years ago, cyanobacteria shows up in the record. And I kind of marked it right here, where on that scale that is, right here. At this time, 3.5 billion years ago, it was just regular cyanobacteria, so not necessarily producing oxygen. 2.4 to 2.3 billion years, it's first rock evidence of atmospheric oxygen. So this is in the fossil record. Here's cyanobacteria evolving right here, shows up in this fossil record, and then it takes a long time before we have a cyanobacteria that's actually has some type of rudimentary photosynthesis going on, which is now forming the oxygen in the atmosphere. 2.4 billion years ago, the first known fungi, and that shows up about the same time that we have oxygen in the air, atmosphere forming. 1.6 billion years ago, we have a Raphitasmia cutensis, which is like a thread-like algae, and Ramothalus lobatus, which you can see on the record right here. It is about the same time when you have the most rudimentary life forms forming in the ocean. So nothing quite on land yet. This is all just water and ocean. And then a little bit after that, 1.2 billion years ago to 720 million years ago, you have the oldest known red and brown algae found in India, according to the fossil record. And then again, 419 to 358 million years ago, Protaxides, and that is the name of the big giant fungi. I forgot to mention that when we looked at those pictures of the big giant fungi that was growing. It's called Protaxides. A terrestrial plant-like fossil interpreted as large saprotropic fungi, possibly even a Basidium micota. And it shows up right up here. Fossil record. And then 407 million years ago is the oldest known terrestrial flora. This is the first known lichen one I brought up earlier. And I think I do, there you go, I have pictures. Okay, cool. So this is the fossil itself, and I cannot tell you up and down on this. I have no idea what is going on in this, but pretty much this is it. So you have this fungi that is formed just like our current lichens, and then this is probably a mixture of fungi and algae within here. That's all I can tell you. I don't understand this structure whatsoever here either. So what is a lichen? Going back to the partners, the symbiotic relationship, fungal partner, the microbiont gives protection. They're mostly ascomycetes, and they're only found in lichens. Photosynthetic partner, the photobiont gives fixed carbon, sugars, green algae or cyanobacteria, and is not free living, sorry, it's not free living. And then just recently, we found a third partner, a yeast partner, obscure basidiomycete fungi called Cyphobasidium. We've known about the relationship between the microbiont and the photobiont for about 130 years. But this third partner, we just found out about it within the last 12 years. And
ascomycetes are compared to basidiomycete.
sure.
maybe just quickly, because a lot of people won't know what an ASCO.
Oh,
is, but
yeah,
to cut
you're welcome
some
to. I usually get into that more, but I deleted it because I don't have an hour.
right?
I didn't really
Or
want to,
the
yeah.
common, like mushrooms versus cut fungi or something. It would just help.
Yeah,
That's
I don't know. Okay. So most of the lichen body is fungal hyphae. Photosynthetic partner exists in distinct layer or could be mixed in. Sexual reproduction of lichens is of the fungal partner only. So let's talk about mutualism a little bit, the types of symbiosis. So just for background, these are kind of definitions. A symbiosis is two organisms living together in intimate physical contact. Mutualism is both organisms benefiting from the relationship. So you can have a mutualistic symbiotic relationship. Parasitism is one that benefits and one that loses. And commensalism is one benefits and the other is not affected. So the big question is, are lichens a symbiotic association between fungi, lichen fungi, and other photobionts? These little cartoons are Clara Schnarga. She's the lichenologist at Berkeley, and I just love her little cartoons. They're really cute. So it shows here fungi. Are they like farming algae? You know, maybe algae is protected. Yes. Is it gaining much besides protection? Maybe not, because it gets its own food from the light. And here's fungi just kind of like farming it and taking it. So maybe maybe it's this kind of relationship. There's also a slaveholder right down here where, you know, fungi is like not only farming, but maybe they're holding them as slaves so that they can get that nutrient, you know, or maybe it is a complete symbiotic relationship. So here's, you know, algae and fungi like, hey, let's get together. I really like her. All right, so metabolism is really, really important to understand in lichens. We use metabolism within lichens to for identification and other things. Lichens produce compounds known as primary and secondary metabolites, also referred to as lichen acids or lichen substances in some literature. There are more than 700 secondary metabolites known from lichens. They're bound to the cell walls and protoplasts. And the most common intracellular products include proteins, amino acids, polyols, carotenoids, polysaccharides, and vitamins. Some of these are synthesized by the fungi, while some by the algae. The majority of organic compounds found in lichens are secondary metabolites of the fungal compound. Laboratory tests have shown that a number of invertebrates will avoid various lichen metabolites, and some lichen metabolites will induce slower growth rates in invertebrates that have consumed them. Some compounds or such compounds give a clear survival benefit to the lichen involved. We don't really know too much about the interaction between insects and lichens. We know that there is something going on. Many times when I look under the microscope from a freshly collected lichen, I'm constantly seeing micro spiders, micro bugs, and I don't know the relationship. We know maybe some, you know, I recorded one little micro spider who formed a little hammock cocoon, little tiny like rudimentary web, and it came out and literally started eating the lichen and went back in. Most insects can't puncture through the cortex because it's so thick. We also know that there's some of these little micro bugs and spiders that will eat the fungi off the soil. What are they puncturing into that cortex to eat it? We don't know. We keep trying to record it and document and specimen the insects, and we still just don't know enough. So very rarely will we find the cortex eaten off by insects, very rarely. So here's some a little bit of an evolutionary symbiotic in relationship with, you know, hiding. These are all insects that have learned how to hide amongst the lichens. Interesting. And then here's a picture. It's a terrible picture, but this shows some kind of insect that has actually eaten on the lichen. Some compounds in the upper cortex of lichen thalli reduce the intensity of light reaching the photobiont cells. For example, photobionts in the genus Trabuxii, which is very common in lichens. One of the most commonest photobiont genera. Function best in relatively low light. Lichens with such light filtering compounds can show variations in the concentrations of compounds, with thalli growing in exposed locations, having higher concentrations of filtering compounds than thalli growing in shaded. So this is a really good example. This is the same exact species, not same lichen, but same exact species. And the one that is very, very orange is exposed to sun, and the one that has very little orange is completely shaded. So you can see the compounds in action with the protecting that algae that's within here. In one study, they enlisted different types of isolated chemicals, such as ooznic acid, uznol, vernic acid, barbatic acid, and orsinol of lichening origin. These chemicals have several bioactive properties, such as antimicrobial, antibacterial, antifungal, anti-tumor, antiviral, and antioxidant. Ooznic acid and its derivatives are more effective than normal medicines in recent reemerging mutant infectious strains. Hence, this medicinal herb may be used as a proven drug in the near future clinical application. Here's the paper it actually came from, which is at the bottom here, if you guys are interested. So pigments and dyes is another type of secondary metabolites. Like works as a mycopigments, algal sunscreen, like I had mentioned earlier with that one species, protecting sensitive algae from harmful UV radiation in sunny habitats. Brilliant yellow pulvinic acid, or the reddish anthraquinones, which are all the orange lichens. Anthraquinone pigments are found in a widespread but scattered range of fungi and plants, but are particularly abundant in the lichen-forming Telochistaceae. Telochistaceae is basically, again, all the orange lichens. That's the family name, where they provide sunlight protection, especially needed when growing in arid deserts of the world. Anthraquinones was found to have evolved in lichens in conjunction with the ecological switch to exposed rocky environments, allowing them to colonize swaths of unexploited habitats worldwide and sparking an acceleration of diversification. So this is my attempt to be like Clara Schnarnagel with her comics. So here you have, you know, a fungi and the algae working together, but then you also have all these protective secondary metabolites that could be several of them, or it could be one or a few in a lichen, depending on what genera. All right, let's talk about morphology. So lichens come in 3 different morphological types. You have fruticose, foliose, and crustose. I keep having this little thing pop up. Sorry, okay, is it gone now? There we go. All right, so fruticose, foliose, and crustose. So let's talk about a little bit about each one of them. Fruticose, the main thing to remember about fruticose lichens is there is no distinct top or bottom. They're usually like round in some way or another. This is a Usnea longissima. It's considered rare up in Oregon. It can grow up to 10 feet. You can see it's really long on this tree. This is my picture up there. And the neat thing about this lichen is wind will come through and it'll drape across, and it's called lady's necklace because of that. But I've seen it really super long, like literally hanging down in the road as I'm driving. They can be pennant, hair-like, upright, and shrubby, or upright, cup-like. Mini fruticose lichens, whoops, sorry. Many fruticose lichens have round branches and have a central core, or others are hollow in the middle. Other fruticose lichens have flat branches that tangle up with each other, or often round in cross sections. Fruticose lichens are mostly two-dimensional structures. Fruticose lichens are three-dimensional, or very three-dimensional. We'll get into those. Can even be bushy or seen hanging from trees. They stick out basically from their substrate. Foliose lichens are leaf-like. Foliose lichens have two easily distinguishable sides. So if you grab any one of these on this picture, flip it over, it's going to have a completely different color and shape of the side. So it has a bottom and a top, unlike fruticose. They can be very flat, leafy-like lettuce, or leafy-like convoluted, full of ridges or bumps, or smooth, hollow or inflated. They adhere to their substrate usually loosely and sometimes centrally attached, or they could be fully attached, like on this upper one. You could see the thallus are really attached all the way to the ends, whereas these are more hanging. Ah. Right. And then you have crustose lichens. Crustose lichens are just that. They're crusty things on rocks and trees. They're pressed either loosely or tightly against a substrate. Sometimes they're embedded in the substrate. They form a crust over just about any surface, such as boulders, rocks, a car, an old car, bark, wood, your roof shingles. They can come in many bright, vibrant colors, sunny yellow, orange, red, as well as grays and greens. Most crustose lichens do not have a lower cortex. So most lichens will have, like if they have a bottom or top, they'll have that strong like skin cortex. They'll have a bottom one too, but crustose don't have any bottom hardening because they're so attached to that substrate, they don't really need one. Crustose consists of 75% of all lichens on Earth. And here you can see these structures right here. These are actually the apothecia. These are the sexual reproductive structures. You can kind of barely make out on some of these right here. Ignore that one. That's not even apothecia, but this right here, these are apothecia right here. So if you were to take a slice of these, which is what we do often to identify them, you can see the spores. So they're always 8 spores, usually almost always 8 spores per asci, which is the sac that it comes in. And you'll see like usually 8 spores. And we usually will have to extract the spores and measure them and determine, you know, what type and whether they're septate or not. Just all the identification things we got to do. All right, so habitat and climate influence, increased air pollution. So they had a study up in Oregon. I forgot to bring that book. I was going to show it to you, but Forest Service is putting out the study where, well, first of all, let me read this. It's as chemicals in the air increase, such as mercury, copper, lead, zinc, and nickel, cadmium, chromium, and sulfur, lichens will go down in diversity in an area. So the higher concentrations of these pollutants in the air, the lower diversity the lichens will be, because they can't really tolerate that. So they had a study going on. They still have it going on to this day. And on the forest I was on, one of the forests I was on in Oregon, we had a couple of study plots out there of this study going on. So what they do is they went in there and they did a complete survey of all the lichens, figured out what species and the concentrations of them. And over time, they would go back and look, and then they would compare that to the stuff in the air that they would find, the pollutants. And they found that there was a direct correlation. Pretty interesting. Air quality monitoring. Okay. So increased fire. This fire occurred in 2018, the Cranston fire. And we had a request from Europe to collect a specific species. So we went out there, didn't realize it was within the fire, so it was gone. But this is a really good example of habitat and climate influence, how, you know, our climate is directly affecting the diversity of these species. We don't know how much unless I could get out there to the site and actually do a study. We know that it's been burnt, so the study can occur anytime. We just go out there, find out what is lost, what was burnt. And then you can even go out to, you know, the outer perimeter of a big burned area. And this is an example. This is the burn afterwards. You could go out to this site right here, go out to the areas where there was still vegetation, do a study on what lichens are there, and then over time go in and see which ones have moved into that area. There's all kinds of studies you could do, but we just don't ever do them. All right, most common. So I'm going to talk about most common species now. Most common mosses of Southern California. Bryum argenteum is one of the most common species of Southern California. You'll find it in your yard. You'll find it everywhere. It's pretty common worldwide as well. And it's also called the silver lichen, or silver moss, because it forms the silver appearance because the leaves are all, it's called hyaline, which are clear. They have no photosynthesis going on. They're completely clear. Funaria hygrometrica, also worldwide, one of the most common ones. There's some growth. They produce these long sporophytes that then turn curly. This is one of the only ones that does this. So if you see this in your lawn, you know exactly what it is. There's only one that does this. Also, I'm not really getting into the spores today, but spores normally will have an opening at the top. These haven't been released for that opening. They still have caps on them. But when these caps are released, this is one of the only families or genuses that will have the hole on the side instead. So it's not a symmetrical, it's more asymmetrical where the peristome opening would be. And you would see them like later as they mature past this.
Is that a fire follower?
No, it's I'll follow you anyway.
Yeah.
It's everywhere.
Often occupied.
Your yard, lawn, everywhere. Grimmia laevigata is one of the most common grimias here. It has very hyaline awns, and part of the leaf, you can almost see it right here, part of the leaf is even hyaline at the very tip here. And then it produces these long awns, and they're hyaline. It's a pretty robust one, robust moss, and this is what it looks like when you come across it in the field. Tortulas and centricheas are very similar, of the same family. And these are the most common ones that you'll come across: Princeps, Ruralis, Vericens, and then Tortula Bryophytes and Moralis. They will almost always have these awns, hyaline awns, but not always. There are a few species that don't have them. And also, they come from the Potaceae family. Let me go back. Rid of that. Potaceae family, and most Potaceae family will have, if you look at it in the microscope on the leaves, each cell will have a papillae. It's like a bump. They'll have different types of bumps on them. Sometimes they're horseshoe shaped. Sometimes they're just bumps. Interesting. Here's some more Grimmia. Some other more common Grimmias, Grimmia pulvinata or orbicularis, montana, and alpestris. Once you see them on them, you'll usually see them like this. You'll see the hyaline awns. Sometimes they'll be all white. They'll have a thick coating of it. Sometimes they won't. Sometimes they'll just have a few. Pretty indicative of Grimmias. Scleropodiums you'll find more in riparian areas or areas where water drains longer. This is a pleurocarp, so it's a well branched one of the branched ones. There's another branched one, pleurocarp. Homalothecium is pretty distinct because when it dries, they always curl up and they always trail. So even so this is a smaller one that will trail a lot as opposed to some of the larger ones, different species, and you'll still get that trailing going on. But again, this is a pleurocarp, so you can't really see it, but there's sporophytes coming off the stem right here. Another one is Orthotrichum. You will see them on rock, but you'll see them on bark a lot. They always have these like hairy caps with little hairs on them.
That named after Denver.
Oh, yes. This one, Norrisii? Yeah, there's quite a few moss named after dead Norris. Yep. Brachythecium, another common one. Brachytheciums are one of the harder ones to ID, no matter how many leaves you look at. So I'm just going to stick with albicans as the number one. Another Orthotrichum one, Lyellii. This one's unique. Once you see it, it's unique because you could see these like little red things on the leaves right here. And if you looked closer, that's what they are. They're papillae. They're big papillae. So even in the dry state, you'll see them. It looks a little crusty, reddish, brownish here. Yeah.
My thesis, Mom.
What?
That was my thesis.
Oh, really?
Yeah.
Anicolia, another common one. You could see it growing in between. There's another moss right here, but it's much bigger moss. Different ones. This is what it looks like when it's dry. Usually does curl up to. In the study I'm doing in San Diego, it's really associated with a habitat we determined as a moss garden. So in almost every moss garden, we're finding this. So Didymonon or Didymodon has broken down into several species names, genus names. If we can't ID it in the field, we just call it Didymonon until we get to the oh, it's Vinea-labrium, you know, then we have to key it out to figure out what it is. So these are just some examples of some different ones. Very triangular shaped. The leaves in particular are very, very triangular. That's a indicative ID for out there in the field. So liverworts, again, we already went through these ones. Asexual, asexual. This is another very common one, Asterella californica. Very, very common everywhere. Produces the archaeogoniophores like this. They are split as opposed to Pomeri, which are very rare, round. As soon as you see that round, you know it's Pomeri. Here's a couple others. Anura pinguis is not common down here. I probably should have taken that out. It is here, but it's rare. I think I found the first one in 2004, but it's been found since then. This is a leafy one right here called Perella bolanderi. And then Fossembronia, the only way to really ID this is to look at the spores, and you've got sporophytes right here. As soon as you see those mature sporophytes, we look. They have these wings on the spores in the microscope, and depending on how many wings you see coming off, we have to count them all the way around the spore to determine what species it is. Right. Cephalosiella divaricata, one of the most common liverworts. I think I also found the first one back in 2004 in Big Bear. But it's all over. It's just nobody was looking for it. That's all. Not that I found the first one, but first one to actually look for it. So this is what it looks like. It's very, very tiny. This is definitely microscopic. But you'll see it like commonly on the ground as these little black things. Sometimes they'll be mixed with moss. They get really dark. They could be like grayish to really dark. But you have to look really closely and then you're like, oh, it's Cephalosiella. Targiona hypophylla, very, very common as well. It's called orbis seed. I don't know why it's called orbis seed exactly, but the involucres are underneath these right here. And when they dry out, when I say involucres, they're like little black balls underneath the thallus right here. And when they dry out, you can see the balls, they form. So even when they're dry, you can ID them compared to like Astrella. You'd be like, oh, it's Tarsiona. I have another picture of it. Yeah, right here. Here's some better picture right here. So underneath that thallus, you'll see these big black things. This one has a lot of it. So again, just a brief, there's the Astrella, Marchantia, Fossambronia, and then the Perella. This one's a Perella, and then Tarsiana. So those are like, I would say, our number one most common liverworts in Southern California. And then most common lichens, we have this one right here, which is Acrospora. Acrospora socialis is the number one most common crustose lichen in Southern California. Probably California. They should have named this one the state lichen, but instead they got this big pretty one right here. That's the state lichen. It's called Ramolina menziesii, which is a net. It like forms like these net structures. Also very common is this Protoparmeliopsis moralis, which is, I believe, common worldwide. This one also, I believe, is common worldwide. This is a Candelaria pacifica. And then we have several different species of this one. Some of them will have this apothecia, some don't. Some will have these like little finger structures coming off the thallus, called isidia. Some don't. So these ones are really hard to ID. This one is really common. This is on trees. This one's also common worldwide. And this one's common down here in Southern California, Umbilicaria fea. There's a couple more. This one's a Candelariella as opposed to Candelariella. And then we also have Latheria. This one, one of the species is called Latheria vulpina, which means lethal to wolves. Supposedly, the pioneers used to poison wolves, but I know nothing about that. I've never seen a paper on it. So this is that same one we were talking about earlier, where sometimes it could be in well-shaded areas. It doesn't get very, if it's completely exposed, it gets really, really dark orange. Also very common, I believe, worldwide too. This one is more of a powder one. It's called Chrysothrix. We've got 4 different species of Chrysothrix here, and it's just powder. Another common one, Rhizofskia elegans, easily identifiable because it easily scrapes off with your nail. Another common one, this is common, I believe, at least in the US, very easily distinguished, identifiable based on the hoods it forms. It forms like the thallus will start going like this, like a hood, almost like a hole in there. I mean, you can kind of see a couple of them right here. And then Usnea is also a unique one. This particular one that turns red is not common down here, but it's such a pretty one. I put that one up, so. It's mostly common on the coast. We have found it in San Diego. Okay, so I have a couple projects. If you ever had any photos and you wanted to identify them, you can submit them to the Bryophytes of Southern California or the Lichens of Southern California on iNaturalist. And usually myself or others that are really good at lichens and bryos will identify them for you. This is the Bryophyte one, and right now we have 173 identified. On my list, I have I think over 600 species in Southern California that have been documented here, but for this project, you could see what area we cover. I thought it was a good idea to kind of separate them out because you have one for California, and California is so different from the North and the South. I mean, the North is almost like you get way up there and now you're starting to talk about the Pacific Northwest, and all the plants and all the lichens and bryophytes also are very Pacific Northwest. And then you have the central location, then you get Southern California, and it's like totally different. You know, a lot more crustose. We have a high diversity of amazing species down here that they don't have up there at all. So that's why I created these projects to kind of document what it is. And the cool thing about this is also you can click this right here on species, and you will get, oh wait, sorry, this is Bryophytes, lichens. You will get this sample right here. So if you click, sorry, if you click species, what it does is it tells you starting from the most common species that are documented on iNaturalist. So right here again, these are all the ones I just covered because these are the most common ones. People keep identifying the Xanthoparmelia cumberlandia incorrectly, so that's not, they just call them all cumberlandia, but there are several different species. But for the most part, these are the same ones I just covered because these are the most common ones and common ones observed in the field on iNaturalist. So this is really helpful. You can click this observation and then you can see everybody's, you get a kind of view of everybody's, observations, and you get to see the different habits and what they look like. So the current project I'm doing is at Torrey Pines State Natural Reserve for the Park Service. We're not doing it for the Park Service, but they're letting us do it. We're doing a complete coverage of all lichens and bryophytes throughout the entire park. We've been doing it for 2 years, and we have gotten through 2/3. So I'm right now re-upping my permit, and this is the last part we're working on right here. And these are just a couple examples of 2 lichens that we're currently working on. This red one was the first documentation in all Western US and all of California. Was first described in Mexico in 2005. And then that was the only known location in the world. And then we found this, so I went up and looked, and sure enough, it was also found at the very bottom tip of Florida. So we just found the 3rd location in the world now. Yeah, well, there's another one that looks just like it. It's called Cinnabarina, Arthonia cinnabarina, but this is not it. And we thought it was that one. We just thought, oh, it's another Cinnabarina, which would also be rare down here. But then when you look at the spores, it didn't. It was like, oh, this is different. So real excited about that one. And then this one right here at the very top, I'm actually currently working on. The experts who wrote the papers on this one, we don't even know what, we're not even sure on the genus, but the genus that we're going towards, the people who wrote those papers are in the Netherlands, Sweden. So I'm in current contact with them, and I just got the key. So today, actually, I'm excited to go work on it because I got the key. So also, all the species of this key are not documented here. So regardless of what it comes out to be, it's still going to be a first occurrence in probably all Western US and California. And this is just 2 examples. We've had amazing findings out there. When you're covering an area like this, and you're going through the entire thing like this, I mean, we're covering everything in there. And the reason this project kicked off was because we had a lichenologist who claimed that a lot of the previous lichenologist who claimed that a lot of the species are going extinct. And I know why he thought that is because we had a lot of collections that were from the late 1800s to early 1900s, and he went out to go find them, couldn't find them, and so he claimed, oh, they're extinct. And we thought, well, why not prove that? Why not get baseline data? So this project is doing a couple things. For one thing, we want to know everything that's in that area. So that's what we're doing. We're compiling a master major huge list. We started out knowing 86 lichens and 0 bryophytes ever documented in there. And we have now added almost 150 new lichens just identified. We're still not even done identifying. And like 65 moss, 20 liverworts, and 2 hornworts. So yeah, and we're not even done. So if we just now found this one because we're in a forested area in this area, this area right here, which is all Torrey Pines actual forested area. The ground has pine needles. It's very forested with canyons. And actually, that's also where we found this one as well. And we've only found it in one spot. We haven't, first canyon, found it, going to each canyon all the way through, haven't seen it again. So it might be that rare, might be very rare. So yeah, I mean, okay, so we're doing to find out exactly what is there. Once we determine what's there, then we're going to determine what is rare. Okay, this is 2 examples of something that's extremely rare. How much we find it from here on out will kind of tell us how rare it is. And then we can also make recommendations to the Park Service on how to protect it and what actions to take to for protections. The second thing is, once we do find one of these, we go and map every single one. So like this red one, we only found it on a couple trees. We're literally going back to those trees and counting every single one on each tree. And we also have another one, which is a lichen that grows on rabbit poo. I should have put that one up here. It's rare. And I had volunteer students underneath the chemise, you know, going through rabbit poo, found one. And I'd go run over and map it. And so we have a complete map of the entire, that population. And the purpose is, in 20, 30 years, people can go back to those already mapped populations, find out if there is a reduction, compare it with the climate. And now we have actual data that says things are going extinct because of climate change or whatever, right? And they can monitor for years. I probably won't even be able to go out there in 30 years. So, but I have a really good crew of young students. So hopefully we'll continue having more lichenologists and bryologists in the future. All right. So that's my talk. Any questions? This is a picture of once a year we do a SOBI free. It's I don't remember what SOBI free stands for, but it's a bryological gathering every year, and every year it's in a different place. This was, I believe, Santa Barbara. So this is all the bryologists. I think Jay Shivak is in there, and
So we
yeah.
do have a couple of questions online.
Sure. Try.
One, Irene Rosen, you might have to repeat this, is asking, will you give us a key for bryophytes? Other references, please.
So one of the questions online was, is there a key for bryophytes? Yes and no. There is sort of a book of California bryophytes. It doesn't have all the species on there, but it has a really good representation to at least get you to the genus. In some cases, if it's common, then to the species itself. It's not a field guide. It's more of a lab guide. So you really need to be able to go into microscopes and key them out. There is a key online available. I believe it's at Berkeley, the Jepson Herbarium is where you'll find that key. So there is a key in there. You really need to have a partner book. It's called the Illustrated Moss, a guide to illustrated mosses, and it's like the dictionary of the terms you need in order to key. Bad news, that book is now out of print. But contact me, I may be able to help. And then for real quick, for lichens, this is the best book for California field guide. It's called the Field Guide California Lichens, and it's great. It's doesn't have all of them because, you know, I think my list has 1,700 species. This is not going to have them all, but it will get you to the most common ones. It also has Northern California, the bigger foliose ones in Northern California, but it's great. It'll get you at least the basics and a better understanding of the genus.
If I try holding up to your face, it might not work.
Yeah, I don't think so.
Oh, it kind of, yeah, just say, oh, that worked. Oh,
Yeah.
but now
I had a slide with these on there. I should have put a slide with
Oh,
both
not
these
showing
books.
that. It's showing it. Oh, that went away.
Sorry.
Okay. Then somebody said, what was the name of the illustrated moss book again? So you can say that again. And then what is your
Striking.
INAC handle?
Oh, Moss Geek.
Okay, so yeah, you might want to repeat the question.
So the question is, what is my INAT handle? It is Moss Geek. And if you post it in those two projects, myself or I have a Jake Bauer. He's an amazing bryologist. He's on my crew, and he's just got accepted into the UCLA program because he's so amazing. And I also have a amazing lichenologist and mycologist, Lauren Yonkers. She goes by Acorn Cap on there. She's amazing. She'll help also ID. And then of course I got Jake all into lichens now. So now he's going from Bryophytes to so he's doing both. I have amazing crew. Those are my core crew members, those two. So if they don't ID, there's also a couple others that was there was a lichenologist in Santa Barbara who was helping me ID a lot of stuff for lichens.
If you can repeat it, somebody said if you can repeat the name of the illustrated moss book.
Yeah.
Maybe I can type it into the chat.
I don't know the exact wording.
Okay.
But it is out of print. How about just have them contact me?
Um,
Contact me and I can help them with that book because the book is out of print.
okay.
And if you were to find it, it's like $900 now because it's so amazing. But I could help. Secret ways.
Chat. So hopefully she's heard the answer to contact you. at
Yeah, so the question
at
was
Mossgeek.
the
Yeah.
illustrated guide. I'm trying to remember the name of it. It's I think it's just illustrated guide to bryophytes, something like that. But just tell her to contact me and
Yeah, I think if you say it, she'll hear you, to contact you at mossgeek@gmail.com.
email Yahoo.
At yahoo.com, sorry.
Yahoo. Yeah,
But you have to say it because she won't hear me.
I did.
Oh.
I'm
the
talking.
whole thing.
Yeah, I just said contact me if about that book and I will help you.
Oh,
Yeah.
say the email address. Mossgeek.
At mossgeek@yahoo.com.
Yeah, it's on the screen.
Yeah, exactly.
Yeah. Okay, great. And
Okay.
then I think we've got, okay, that was answered live, I'm looking for if there's any more in the chat. I have a question.
Okay.
You'll have to repeat it. That red moss that was so rare
Mhm.
that you found, was that on a Torrey pine or a different, like a hardwood?
Yeah, it was on a lemonade berry and it was also it was on a couple other things.
Interesting.
Yeah, and we're not done mapping it, but we gotta go back and finish counting every single one.
Yeah, so I'm just wondering about
Mhm.
dispersal, potential.
So here's the
dispersal.
thing. There are identifications of the Cinnabarina, which looks almost exactly like it.
Uh-huh.
We're wondering well, are people not looking at the spores?
Yeah,
That could be a possibility. It could be underlooked,
yeah.
basically. Yeah, and then the other one, this top one here, I found like 4 or 5 sites of it. And again, nobody's going into areas, we're weed whacking into some of these canyons. We're way in there. We're trying to get every single type of habitat in there. You would never see that if you didn't do that, so could be another under collected. Because a lot of the lichens, like I have a record of all the people who went in, all the people that went into Torrey Pines starting from 1920 something. And some of them were just like botanists. Some of them are just like, I'll collect 1 or I'll collect 5. And then finally the last one, which is a world-renowned lichenologist, Carrie Knudsen, he went in there and collected a lot. And by the time all 6 people made a collection, we had 86. And all they did was go poking. They weren't doing full surveys like we're doing. So we go every other week, and it's, like I said, we've been doing this for 2 years now, and you see how much we've covered.
We have somebody that's trying to ask a question, Denise Mitchell, and I'm asking her to please type it because she won't be able to.
Oh,
type it.
so Denise Mitchell, who is trying to ask a question, please type it because you
Yeah.
can't hear. her?
We can, yeah.
Oh,
So
is her
I'm-
mic off?
I'm
maybe?
just sending her that.
Oh.
So, yeah, so maybe she'll type it in. If she types her question in. We'll see if we get
Got a
it.
question?
Yes. I actually have two, but let's start with one. The horrific fires we've had in Southern California.
Mhm.
You mentioned air pollution as a detriment to healthy lichen growth and other
Yeah.
plants. But what
Particulates.
about soot that lands on the soil, that lands in the trees, that lands everywhere?
Those are actually healthy for the
I was
environment.
yeah. Is it good or bad? Yeah.
Yeah, they're fine. They're not like, I don't think they have an effect on lichens because if they temporarily land on there, lichens are like moss. They can completely desiccate. I've pulled out specimens in my hot garage for 10 years and hydrated it comes right back to life. So it's just a matter of the suit temporarily, in dormant stage, not able to photosynthesize, but they don't die from it.
It might depend on how close they are to the toxic ash that's coming from not vegetation, but from structures.
Structure, yeah.
Yeah, so when structures are burning, you get a lot of really awful stuff.
Sorry, I was asked about
Yeah.
fires and soot landing on species.
So, but in the natural environment.
It adds a lot of, a lot
Yeah.
of new nutrients and carbon to the soil.
Yeah.
Mm.
I knew that, but I was just concerned about I didn't explain it well enough, the fact that it's, you've got not only wood burning and plant material burning, but you have trees, houses burning and infrastructure burning and, all that provides some too.
Right,
Mhm.
that, yeah.
Yeah.
From that can carry some.
So another benefit of our surveys is the fact that a couple things. So we have collections. So what if that canyon with this red lichen goes up in flames and is a total loss? Well, once they restore it and plants start growing in there, we can probably take our collections and add some spores back out to the thing. So that's a benefit. We had a lichen that we were looking for that was documented in 1984. That was the only known Ramalina peruviana, which is in Peru and South America, some in Mexico, and then nothing here. That was the only known site in all of Southern California. So we were looking aggressively where it was collected, and there was a lot of Ramalina there, and we're still not even sure if we collected it. But that was another example of, if it was a shrub land. So if that went up in flames, we have collections of a lot of species that we can reintroduce back in there. And not only that, but just knowing where they are helps the Park Service to really focus, like definitely protect this site right here, because we have this extremely rare red lichen, only known on these couple trees right here. That's it so far. We're still working down, but we haven't found it again. So
I'm gonna bring. So here's Denise's, if you could just read that. To people.
So Denise Mitchell says, study environmental factors. I have the pleasure of being at Big Sur and saw an amazing display of lace lichen among the coast redwoods, foggy, Point Lobos, State Reserve. Is it Lobos? Okay.
Yeah.
Curious if I would ever see what kind of display in more arid environments. Also, I worked with Lauren in Santa Margarita. Lauren's awesome. I'm so stoked to have her and Jake and these amazing young students who are just becoming fantastic future, hopefully.
So Denise is the one that I think is gonna give our April program. I have to get back to her.
So yeah, Big Sur and those coastal areas are just amazing. They have a lot of amazing species up there. Totally different from down here though. And the lowest known for that lace lichen, we actually have just in Southern California, it seems like the populations are all coastal, all the way up from Oregon, all the way down, and then they start breaking down and you don't see them. And then Lauren just found some down here somewhere. It's like literally 2 locations in Southern California, and that's it. So we haven't found any on the coast. Question?
So, with respect to fossil bryophytes, are there any sites in California where you can find fossil bryophytes?
Not that I know of. The question was, is there any sites in California for fossil bryophytes? Yeah, not that I know of.
So the other question is, kind of very ignorant question on my part. On the crustose lichens that you see on the granitic rocks,
Mhm.
are they only metabolizing when there's a rainfall?
Yeah, if they go dormant. Right. I take that back. No. Because algae is constantly alive. It takes a lot to really, really dry out completely. Once the algae dries out, the lichen's dead. I mean, it's a dead lichen. It's all just fungi at that point. So the fungi is able to keep those algae completely hydrated so that they can, you know, photosynthesize and produce the sugars and the carbohydrates. So, for a long period of time. I don't know. I don't think that they can come back once they're completely dried out.
A
Yeah.
second question.
Another question.
At the beginning of your talk, you mentioned how algae, fungi and such move from the ocean to land,
Yeah.
but they still reside in the ocean.
Yes.
And one of the reasons I found it interesting is they provide food for fish.
Algae.
Whales, for instance,
Uh-huh.
take in algae. That's their primary source of nutrition.
Yeah.
Is the algae in the ocean.
So those are different types of algae. The evolution of different species occurred, the big radiation of adapting to new environments. So the bryophytes evolved from coastal mud flats. I don't know if they were freshwater or not. I mean, they must be related to the ocean because it was algae that was evolving onto land, and they had to be able to desiccate, like dry out without dying, right? Yeah, so, and that's the first land plants that we believe how they evolved.
My background is in nutrition and biology. One of the more common supplements that people take is fish oil capsule because they're high in omega-3 fatty acids, which is beneficial in some regards. That comes from the fish itself, but the fish get that from algae. That's where they're getting that from.
Interesting.
Yeah.
Peter says that his
laboratory-
background is in plant
laboratory,
nutrition.
They're now creating the same thing in a laboratory form, called algae oil, in which.
Talking about
in which
fish oil
you
and algae
consume
oil.
You consume it instead of fish oil.
Mhm.
So there's a beneficial bacteria, there's a beneficial nutrients in algae, nutritionally.
So I've been finding moss poo. It looks like some insect is eating the moss, and then like there's little poos full of eaten moss. I have no idea what insect is doing this. I just keep documenting it. It would be awesome to find out what insect is eating moss. That's incredible to me. But I don't know. Yes?
What do you know about tardigrade?
I love them. So I was asked about tardigrades. One of the SoBe-free events we went to, we had a tardigrade expert show up from Berkeley, and she showed us a trick where you could take a bundle of moss that you've collected from the ground, put it upside down in a petri dish with water. The next morning, take it out, look in the microscope, and there's tardigrades there.
Wow,
Yeah,
they call them water bears.
yeah, yeah. Question?
Yeah, I had a question. When you were talking about the very colorful crustaceous lichens, not like that one, but the ones that really now is that typical the strong color and the lichens that are having sunscreen?
Yeah, that was that's what they're believing, that you know, as a secondary metabolite, those arthoquinones are being used as a sunscreen to reduce the radiation that might be harming the algae.
I have another question unrelated to that, but all of these plants I've loved for a long time, but hornworts I don't know very well. And where do you typically find them?
Here and there.
I mean, they have a certain habitat, I guess, if there's a certain habitat, they're more likely.
So I was asked where I'd find hornworts, and hornworts, they're on soil. So anywhere that's going to have water that's being held longer. Yeah, I don't know. I don't find them that often, so it's hard to describe where you'd find them. In San Diego, we had a specific type of hornwort that was unique for San Diego, which is different than the hornworts we were finding. Say, there's a spot next to near Pachanga in that area, and we found a hornwort there. It was a totally different species than what you'd find in San Diego. And then there's one we found in one of the canyons. I could give another talk because there's some amazing habitats around here. That the canyons in San Diego, we have these big, tall sandstone canyons that are very, very narrow and just packed with moss, bryophytes. It's just amazing. And we found a hornwort in there. Haven't identified it because we couldn't get the sexual reproduction. We got to look at the spores. So we have to go back to that site. Still don't know what it is. And I think those are the only places I've seen hornworts lately. They're not that common.
We've got all the questions, we've got all these questions online answered.
Oh.
And all the ones that were in the chat.
Great.
So anyway, this is great.
Great.
Thank you.
So as the representative for the Bryophyte chapter, I was talking to Arlie about maybe giving updates on a regular basis of other stuff that's going on within the Inland Empire. Unfortunately, I'm not doing much work in the Inland Empire. I still have a gate key for the Forest Service. I'm allowed to go up anywhere because they like it when we go up there. And when I have time, the San Diego project's kind of taking time up, but we still like to go run up there and do a lot of work. I'm also monitoring an undescribed liverwort up there in Big Bear. We still haven't described it yet. It's the only known location in all of California. And we thought we might have had it identified. It is a Fossambronia, but it's submerged completely in water in a meadow, spring fed. Found it back when I was working up there at San Bernardino National Forest, doing the fen inventories. And it looked very similar to one that was found in Argentina and Mexico. Those are the only two other locations of this other one that it's very closely related to. But we found that it's not, we don't think that's it. So we're still looking for the sporophytes because it won't sporophyte. So I started going up there on a regular basis to try to catch the sporophytes. I've even collected some, put it in a little mini terrarium, and I still have it growing. It's growing great. No sporophytes. So as soon as we get the sporophytes, then we can start turning around and I actually identify it to species.
So if it actually rains this year, do you think you'd be interested in leading a field trip?
Yeah. Oh yeah. Yeah.
So we can give your name and contact information to Michael Wagner, who's going to be organizing our field trip.
Michael Wagner.
Mhm.
Is it
Yeah.
somebody I don't know?
Wait.
I was
Yeah, I
just
think
Chris
it's
Wagner recently, so.
Right. I think it's Wagner. Sorry. I'll correct. I'll give it back. I'll connect you in an email. So I get it all
Sure.
right.
So I was asked about giving hikes, and yeah, I give hikes, and I also would probably my crew members would be happy to join as well. They're very helpful. Yeah.
Okay, well, thank you very
Question?
much. Oh, there's no more question.
Have you ever, been out to Hartford Springs?
Where?
Hartford Springs Park?
Harvard?
It's-
Yeah.
Yeah,
Where is it?
it's near Lake Matthews.
in the Gavilan.
Just for the Gavilan Hills.
No,
Amazing, and mountains, different colors of lichens, different shapes.
really?
Some of them.
So it's in the
are
desert?
plants, some on the rocks.
Yeah, on the, a lot of some of the old shrubs. And then a bunch of it burned last year.
Mhm.
And so it was closed last year. That, probably.
It's open again
open
now.
that, yeah.
Huh, is that local? Like where
Yeah.
Lake Matthews is,
Up by Paris.
oh,
Yeah,
okay,
Hartford Springs Reserve, so it's in the
Hmm.
county park system,
nice.
and it's really easy to get to.
So I know, I could easily get a permit with one of the Riverside entities. The other one, I think RCA, they just won't give me one because I don't have insurance for people to go out and collect, which is kind of weird. I was like, I've been out in the woods all my life,
Yeah,
but yeah.
yeah, we can talk about that later.
But at the same time, I mean, in all honesty, I don't have time to commit to another site at this moment. Because I really want to get this Torrey Pines done. And then we have several other parks with the Park Service, all in San Diego. There's several other parks. We're gonna head up even after this. Because San Diego coastal areas are just amazing with the diversity. So we want to really capture that before, all this fires occur and more climate change occur. And we really want to get an idea of what exactly is there and what's rare so that we can protect those.
Any other questions?
Any more questions?
Yeah,
Oh,
one-
yeah,
Yeah, every year, when my kids study biology, they go through primary and secondary succession.
Hmm.
I always get so I'm just sharing this, and how does the lichen actually make soil? And I'm like, I don't have that depth of knowledge.
yeah.
Like shed any.
I don't think it makes soil. It can hold water longer for soil to accumulate.
They can compose and
Yeah.
become part of-
I mean, we found like lichens that are rock specific, but then they kind of move over to soil. And if on some rocks, you'll see where soil just accumulated around something that's growing. And then all of a sudden now you have soil there. And then you have another thing that takes us, oh, there's soil here, and they grow and yeah. So I would say, as far as that goes, it's more of, lichens and bryophytes will hold water longer. So how does that affect, that
makes
dirt
more
spot or
sense
that
then.
rock or that tree? So,
Okay,
all right,
so I think Katie, tell us you can kind of stop recording. We just
Okay.
thank you.
all right.
Thanks. Okay. I can. I think did she stop recording?